Hello, everyone. My name is Raeana, and today I'm going to be talking about human enhancement. So, the question I'll be discussing is, should somatic cell engineering to alter human abilities be disallowed in the US? So, what is somatic cell engineering? A somatic cell can be defined as diploid cells of a living organism that are not reproductive cells, which are sperm and eggs. As you can see in this diagram, some examples of somatic cells that can be genetically altered are muscle and skeletal cells, blood cells, skin cells, nerve cells, fat cells, and stem cells. As far as the process of somatic cell engineering, it includes the replacing of a target mutated gene with its cloned wild type gene, or some gene that has been modified. This edit is widely made using the CRISPR-Cas9 system, where a gene is essentially knocked out of a region of the genome and a new gene is reintegrated. This new gene is brought into the cells, most commonly using a viral vector. However, there are more delivery methods. And this can be done in vivo, in the body, via blood transfusions or IV injections, or ex vivo, where your cells are taken out, worked with, and then put back in. I want to briefly distinguish the difference between somatic cell engineering and germline engineering. Somatic cell engineering deals with the editing of adult human somatic cells, like those that I mentioned in the previous slide, whereas in germline editing, the cells that are edited are reproductive cells or embryos. In somatic cell engineering, the modifications are not passed down to the next generation, so they stay within the individual. Whereas in germline engineering, the modifications are passed down through generations. So let's take a look at what human enhancement looks like. To piece together some sort of definition, I would say that human enhancement involves changes to the genome that boost human capabilities beyond what is considered normal for either the individual or humans as a whole. The big debate with this is what we all consider normal as a society. And this is where a lot of the ethical and moral dilemmas come from regarding genetic engineering. Normality can often be a subjective idea. However, in this case, normality is realistically a range or spectrum, not so much an ideal state. So, therefore, any enhancement can be classified as such when it surpasses this range of normality for a given bodily function. There are a few different applications of genetic engineering regarding enhancement, like enhancement to the physique, where you're improving muscle size or strength, or disease prevention. For example, how synthetic blood can be used to increase oxygen affinity that will ultimately lead to improved immune response and even improved endurance. So now let's look at what the current regulations are in the US and globally regarding genetic engineering. If you look at this chart here on the right, we have a range of 0 to 10 and similarly red to green, where 0 and red both indicate prohibition of any kind of genetic engineering. So we can see that the US, Canada, and Mexico are prohibiting germline editing, and Argentina and Israel have high regulations for that set. As the chart gets into the yellow and greens, that indicates less and less regulation. So we can see that Russia, Ukraine, China, Israel, and Mexico have fairly light regulations on therapeutic genetic engineering. Shockingly, Japan has pretty loose regulation on both therapeutic and germline editing. So looking at the US in particular, they have pretty tight regulations on therapeutic genetic engineering and even are prohibiting germline editing. The main purpose and use of genetic engineering right now in the US is for treating genetically inherited diseases like sickle cell or cystic fibrosis. So there's currently no elective enhancements going on right now. In the US, all research and any clinical trial proposals regarding genetic engineering are required to be approved by the FDA, the Food and Drug Administration. Through the FDA, investigational new drug applications are reviewed as well. The Institutional Review Board approves any clinical applications, and then public discussion is also taken into account, and this is provided by the NIH's Recombinant DNA Advisory Committee. So now I'm going to jump into my pro and con arguments. And to clarify, my pro arguments are in favor of disallowing somatic cell engineering in the US, and my con arguments are against disallowing somatic cell engineering. So, my first pro argument is that there is a risk of incompatibility when it comes to editing the genome. Genome editing is dangerous due to the many risks it can pose to the human body via the body's immune response and potential for additional harmful mutations. One of the most common delivery methods of gene therapy when delivering a CRISPR edited gene includes the use of a viral vector. So, with the use of viral vectors comes an increased risk of heightened immune response. Since this virus is a foreign entity, our innate immune system is triggered in order to prevent an infection. This heightened immune response could impact the compatibility of the new gene into the genome, as it could affect the success of the delivery and lead to rejection of the new integrated gene. CRISPR itself is also not 100% accurate and is known to cause risk of targeting additional cells that can trigger other harmful mutations and even cancer. My second pro argument in favor of disallowing genetic engineering for human enhancement is that the accessibility of this process is limited to a group of people who are financially eligible. Gene therapy treatments cost around $1 to $2 million per patient, so this leaves accessibility to this process to those who can afford it. You would think that if human enhancement were a common practice in the US, taking into consideration how expensive one treatment is, there would be a noticeable socioeconomic divide between wealthy and enhanced and non-wealthy and normal. So, taking a look from a global perspective, this process also requires government funding for things like research and clinical trials. There are limits to this as well, where high-income countries are going to have more freedom in this area compared to lower-income countries. As you can see in this bar graph, higher income countries like North America and Europe have more access to gene therapy trials, whereas lower-income countries like Africa or South America have little access to gene therapy trials. This now causes even more division, where certain countries are going to have advantageous human traits when others are not. When several countries come together, for example, in worldwide sports competitions, do you think that this would fare well? This brings me to my last pro argument in favor of disallowing human enhancement in the US: unfair advantages. With resources available for human enhancement, things can get out of control. What enhancement will do is provide some people, as I've just discussed previously, some people referring to those who are both financially and physiologically compatible with the process of genetic engineering. It'll provide these people with above normal disease prevention capabilities, above normal cognitive abilities, and above normal physical abilities relating to things like muscular strength and endurance. If we look at the bar graph to the left, we can see that the majority of participants in this survey are more worried than enthusiastic about these enhancements. This is due to the many potential health risks, but also due to unjustness and bias. These are hefty resources, technologies, money, and time that are used for enhancement, which would be deemed elective treatments, where those resources could be used for something else, maybe something that will save lives rather than enhancing them. Another worry voiced by the public is that as we go further along the route of enhancing humans, the possibility then arises for the creation of a quote unquote superhuman race, which sounds thrilling from afar and makes a really good storyline for movies, but integrating that into our reality is more of a daunting idea than we think. So now I'm going to move on to my con arguments. And just as a reminder, the con arguments are arguing against disallowing somatic cell engineering for enhancement purposes. So, in other words, these arguments are going to back the idea of allowing this in the US. My first con argument is that the practice of genetic editing will provide the opportunity for scientists to understand the human body and physiologies at such a level to advance related technologies to make them more efficient for further research and application. Some of these applications can include improving CRISPR for gene editing, but also other technologies that can be used for exploring new avenues of human enhancement. For example, with current knowledge, scientists were able to develop a brain chip to restore a man's paralysis in his arm. Now, while this isn't a direct benefit of genetic engineering itself, it is evidence that novelty technology will continue to arise from stepping stones that scientists have left previously. Think about all the advancements that could arise years down the road if scientists were able to conduct research and trials, editing genomes of participants to enhance their abilities. My second con argument is that the altering of human abilities can benefit humanity by enhancing physiological attributes. One of society's many goals is to push for enhancement to improve things that we deem as deficient, like vision or hearing loss. There has even been a push to enhance things like strength or cognitive abilities with the use of substances like steroids or Ritalin, for example. The movement of genetic engineering will provide a way for individuals who desire these enhancements to get them and even evade having to take substances. That brings me to my final con argument for why human enhancement should be allowed in the US is for the enhancement of health, more specifically disease prevention. A major example when it comes to disease prevention is the implementation of artificial blood. This artificial blood is made by modified hemoglobin protein or sometimes with synthetic material like perfluorocarbons. Blood cells can be genetically edited to increase the amount of oxygen it carries, which will aid in preventing disease and increase the body's ability to fight off infection. When oxygen levels are higher in the blood, it allows for neutrophils and other important immune cells to function as they should in immune response. It's also possible for artificial blood to be made without impurities, like disease causing agents, like viruses or microorganisms. If genetic engineering for enhancement purposes were allowed in the US, the use of synthetic blood and other applications can be available to individuals to enhance their health in these ways. So, now that I've discussed a little about genetic engineering and human enhancement, I think an interesting thing to think about is if scientists are working for the benefit of society, as well as the benefit of their scientific community, would working towards human enhancement really be worth it? So, that concludes my presentation. Thank you all for listening, and I am looking forward to discussing with you all in class.